Hello, Professor Faith back again. So this is the microscope lab, lab number 2. So if you have your lab books already, we're going to go through this. There are several videos for you to watch, but I also want to kind of go through this with you by myself. So the first thing you're going to want to do is, again, vocab. So on that first page, there are a ton of vocab words there. Make sure you know them. For sure, I would definitely go through and make sure that you have the first part of this lab. The first part of this lab is naming parts. And yes, that's something that you can easily Google or look at the diagram while you take those assessments. But more importantly, you need to know how, when I tell you to put your slide, you put it on your stage, you need to understand what I'm telling you. You also need to know the function of the stage, which is the whole of the specimen. So you need to know both the structure and the function. So I have a picture here. I just put kind of a crude little picture of a microscope. You have a better one, which I couldn't find online, but you have a better one on page 14. That is the actual microscope that you have used at CLC, the one with the red handle there. So you need to make sure you kind of go through those parts. You need to know what they do. You will be using those in your virtual microscope lab. So when I talk about these things, you need to be able to describe them, especially if you have problems, and then you're not going to be able to tell me what your problem is with that program if you don't use the right vocab. The biggest problem I found with the virtual microscope, specifically the first link I have that uses the Adobe Flash Player, is that notice in the picture here, the objective is in like the front of the revolving nose piece. In your program, it's actually in the back. So like I was getting really frustrated because I could not find my letter E until finally I realized that it's kind of reverse. So do make sure that the 4x objective, which is your scanning lens, is to the back on that particular microscope, or else you are not going to see anything and you'll be sitting there forever getting frustrated. The next thing on page 15, they have this nice little bullseye here, and that is your field of view. Now, the field of view is the area you see. So when you look through the eyepiece, so when you're looking through your microscope, our microscopes are binocular, meaning they have 2 eyepieces. When you look through there, and your virtual does too, and you got to get them lined up. So when you look through your microscope, you're going to see a circle. Now the circle, if you look at the stage has a circle opening, and that's the whole point of a light microscope, is that when you look at a specimen, light has to pass through it. So you do need to have your microscope turned on or you will not see anything. And so you'll turn your microscope on, the light will go through the stage, and then you will see this field of view. Now, notice with the 4x objective, your field of view is much bigger than your 4x. It's much smaller. The circle is much, much smaller. So what we have is this observation that as magnification goes up, your field of view goes down. They are inversely proportional. So again, as magnification increases, field of view decreases. So right here we see our specimen. We see our field of view in this particular example is about 1.6 millimeters. Right, so that's an example of field of view. Now the first thing you would have done is you would have looked, and it looks just like this, the letter E. It's already made on a slide. So as you look at your slide, one of the big things is, and if you can, and I'm like trying to look up real close, I can't tell on that picture, but when you look at, actually my procedure's off, that procedure should be 2.1 still. When you look at the slide, just with your eyes, you will see a letter E. When you look through the microscope, what's going to happen is it is going to get flipped. And so I always refer to the microscope as being backwards world. All right, everything is backwards. So as you can see in this picture, we can see this E is in the proper orientation. Over here, it is backwards and upside down. So it's completely flipped and inverted. And you would have noticed that in the lab. Now, what you will also notice is that when you put, so it asks for questions on page 16, it tells you how is the E oriented. So I would copy down what's on the slide. You can see how with your eyes it looks this way, and with the microscope it's the opposite. And then it asks you to, it asks you to do something, but you really can't get this experience through the virtual lab because you can't really see the stage moving as you're looking through it. So what you would have noticed when you put your E on there, if you move your E to the right, so you're moving the stage to the right, your E will actually appear to move to the left. Everything is backwards in the microscope. So if you moved your E, I think it asks for you to move it down. So if you move your E towards you, the E would actually move away. So you have to get used to using the knobs on the mechanical stage on the opposite direction than what you want it to go, because it will move in the opposite direction that you are moving your stage in. So it kind of takes a little bit of time to get used to it. And then the next question, so if we are here in the proper procedure 2.2, it asks you about magnification and focusing. So when you do this, your total magnification, okay, your total magnification will always equal your objective times your objective times your ocular. So here, if we have our scanning objective, which is 4x, and our ocular and eyepiece is the same term for the same part. I'll use both of them. So in this case, we're using eyepiece. So our eyepiece is 10x. So you're going to multiply those together. 4 times 10 gives me 40x. That's your total magnification. Now the 10x, so we go from our scanning. Our scanning is called the scanning because you can see the biggest field of view, so you can scan the whole slide to find your specimen. Once you get your specimen, you center it on your field of view, and then you bump up to the next objective, which would be your 10x objective. So once you move it up to the 10x objective, then of course you have 10x times your eyepiece, which is 10x. So that's 100 times magnification. Again, you'll center it, find focus, and then we'll move up to the 40x objective. Now the 40x is the high power. So with the high power, we are then magnifying 400 times. Oh, excuse me. So that is how you calculate total magnification. So then number 2 asks you, so you're going to do your math there. Then number 2 asks you which objective magnifies the letter E the most. Well, you got 40, 100, and 400 times. So it is your high objective is going to magnify the most. Does this objective provide the greatest or the smallest field of view? So again, if you go to your bullseye sheet, that 40x gives you the smallest field of view, which is why you do not want to look for specimen, even if it says observe this specimen at the 40x objective or the high power lens. You do not want to start at that, because again, you would be looking through this whole screen like if I was looking through this little hole in my hand. Your field of view is very small. That means you're seeing only a very small part of the screen at one time. All right, so if as you go down in magnification, so you're 10x, okay, my field of view gets bigger and I can look at my whole screen here, and I'll find it quicker than this, all right, but my scanning would be huge, and so I can quickly find my specimen in the screen here. All right, so you always want to start with the scanning and then work your way up to your final magnification. So you go scanning, low, high power. Now, when we look at our E, I'm going to go back one here. When we look at our E, we can see here that with my scanning, right, that's our total magnification here, 40x, I can see the whole E. All right, I have a big field of view. But as I go up to the high power, I can only see a part of the E because again, my field of view got smaller. I'm zoomed in on that E. So again, it's magnified. So magnification goes up, field of view goes down. So you will not be able to see the whole E once you get past that. Even the 10x, you can't quite get all of it in there. All right, now the last question here, number 3, you will notice that on your virtual, they'll actually talk about that in one of the virtual programs, and that as you magnify up, your light intensity tends to go down. So the background light gets dimmer. So there are 2 ways you can correct for this. On the base of your microscope, you have what is called a rheostat. It's kind of like a dimmer switch. So you can turn the brightness up or down. Just wonder what my dog is looking at, up or down on the light source. You also have what is called a diaphragm, a condenser diaphragm, and it's a little aperture that sits underneath the stage. And so that will control how much light actually goes through the stage. So there are 2 places you can control the light. And that's one of my biggest problems, that I have students that get headaches because they've got the light just blasting at them. And I'm like, turn it down and we'll mess with that. So of course we won't have that experience here, but when you do take your next class, if you have to use a microscope, remember it may be too dim, and so you need to mess with those 2 adjustments, or too bright. So those are 2 places you can adjust the light. All right, procedure 2.3 is our field of view. This is probably the hardest part of the lab right here. All right. So what we would have done is we would have picked up a slide, and the slide would have actually looked like there was nothing on it. A lot of students said, there's nothing on it. Yeah, there is. There is a microscopic ruler on there. It's called a micrometer. It is very, very small. If you think of what a millimeter is, if you think of a meter stick, those are the little tiny lines on your meter stick. Well, this is just two of those little lines. And then we can see in here, these are called micrometers. So we're going to be, there are 1,000, 10, we can go 10, 20, one when we count through here. So all the way up, there's 1,000 micrometers in a millimeter. So we could go ahead and count those by, I don't know, count by hundreds or whatever, by 100s. So there you can see all the little lines in there, 500,000. So there's our micrometer. Now, what we would have noticed is that I always call this as like the Goldilocks syndrome here. So you put the ruler and you start again with your scanning. And what you would have gotten is like this. If I was to look at this little ruler on my whole screen, it's too small. I can't measure my screen size. Right. So if we put it on the scanning, we'd have been able to find it, center it, and focus it, but it would have been too small to measure your field of view. And then if we would have went to the high power, it would have been like too big. You'd only see like 2 lines. And so you can't really measure with that. And so then we go to our low power, and it's just right. And so what we are able to do is we're able to see our field of view. We're able to measure that on our low power lens. So you can see on page 19, we have a table. So with that table, the first thing you're going to fill in is the low power. That's the 10x objective. And we would have actually physically measured that. And when we measured that, we would have measured it at 1.8 millimeters. So we would have had these nice little measurements in here. Just gonna kind of throw my fake ruler in here. So we would have had this measurement. It would have been 1.8. We would have done this lab we would have done mostly together anyways. But it is 1.8 millimeters across. Now you have to convert that to micrometers, okay? So what I will do, and I always show my students this, now a lot of them choose not to listen or learn how to do this. I highly suggest you learn it. You will need it if you're going to take physics or chemistry. Most of you are definitely take chemistry. So this is called unit analysis. And so if you take your number 1.8 millimeters, and make sure you put your units in there, and you're going to put it over 1, because we know if we put any number over 1, it's still that number. 20 over 1 is still 20. 4 over 1 is still 4. Now I'm going to multiply this also by a factor of 1, and everything we multiply by 1 is also that number. So the first thing I want to know is, I want my answer in micrometers or micrometers, however you want to pronounce it, or microns. So in order to get rid of my millimeters, I need millimeters on the bottom, and I want my answer in micrometers. So I'm going to set my equivalency up like this. We use a for micrometer, okay? The problem I have here, because notice my millimeters will cancel out, and that leaves me with the unit I want, okay? The problem I have is I am not multiplying this by 1 right now. 1 micrometer is not equal to 1 millimeter. Just like $1 bill is not equal to 1 quarter. 4 quarters are equal to $1. So I have to make this equal. Now I know that micrometers are much smaller, so I need a lot of them to equal my millimeter. And in fact, we have that conversion. We know there are 1,000 micrometers in 1 millimeter. So in order to get that, I'm going to multiply everything on the top and multiply everything on the bottom. If you are not familiar with how to do math with fractions, that is going to be a problem, because we are going to be doing a lot of fractions come the end of the semester. So my answer is 1,800 micrometers. Now, I have a lot of students who don't set up their unit analysis this way, and then they end up moving the decimal the wrong direction. Instead of multiplying by 1,000, they divide by 1,000. But if you do your units correctly, you would have known the units wouldn't cancel out that way. You would have ended up getting like something really silly, like millimeters squared over a micrometer. So you really need to work on making sure your units are correct. Now, what we're going to do next is on page 18, there is a formula. I would highlight it. Okay, so that is the formula we are going to use. So what I'm going to do is I'm going to do it in 2 different colors. Now, since I have physically measured my field of view with the 10x objective, I know one whole side of that formula, okay? So this is an equivalency. What I'm saying is the field of view of one objective and its total magnification is equivalent to the field of view and the magnification of another objective. So in this case, the first one we need to figure out is our, I use different color, is our field of view, because it's asking you, what is the field of view of your 4x objective? Remember, the ruler was too small. So two different colors. Remember, the ruler was too small to measure the field of view in your 4x objective. So what I'm going to do is I'm going to use some algebra. Now, I know my field of view for my 10x. We measured that at 1.8 millimeters. I also know my total magnification, because my total magnification is going to be equal to 100. Okay? And I'm going to put a box around that. On my other side of my equation, so this is what I have. Okay, so I have my 1.8 millimeters times my 100. Now on the other side of the equation, I do not know my field of view. That's what my unknown is. So that is what I am trying to solve. So I am going to call that my, I'm just going to call it F. F for field of view. That's my variable. But do we know the total magnification of my 4x? Yeah, it's 4 times 10, right? The ocular. So my field of view is 40. So right now with my equation, I only have one unknown. So I can do this math. So on my left side, I'm going to take my 1.8 times 100. Again, in this class, I would definitely recommend a calculator. Even though you have your phone, sometimes it's not as quick as having a calculator right there, especially when we will be doing some square roots. All right, so it gives me 180 equals F times 40. All right, so I'm going to do this one step at a time because not everybody in this class is as comfortable with math or algebra as others. Now, hopefully you should be, and some people haven't had it in a long time. All right, so I'm going to divide both sides by 40 to get F by itself. So I just need to divide my 180 by 40. And that gives me 4.5. So 4.5 equals F, which is my field of view at 4x. So you're going to write that in there, and that's millimeters, right? So we did all this in millimeters. So you're going to put that in your chart. So for your chart where it says scanning 4x field of view diameter, it is 4.5. Now you need to convert or multiply by 1,000 to get your field of view in micrometers. Now you're going to do the same thing for your 40x. Remember the 40x, the ruler is it's too magnified, right? I only got like 2 lines, it's huge, all right, because it's really zoomed in there. But again, I know my math for my low power. So again, you're always going to keep that left side of your equation, okay? So I'm simply going to fill that back in, okay? So the same thing. That side does not change. It stays the same. That's the one we were able to physically calculate. That is what we're going to use as our standard. Okay. Now the other side is going to change a little bit because I don't want my field of view at my 4x. I want it at my 40x. And I also want my total magnification at my 40x. So my field of view is I'm going to keep that as my F. That's my unknown. I don't know that. That's what I'm trying to solve for. But my total magnification at 40x is 400. So again, I have a single variable equation, piece of cake. I can solve it. I'm going to go ahead and do this in one quick step. So actually, I'll do 2 quicks. I'll do 2. So we know that we're going to get that 180 again on this side. And then our F, wrong color. And then we're going to I'm going to go ahead and move my 400 over here, right? So I multiplied this, got my 180. I divided both sides by 400. And now I'm going to end up getting F equals, I'm just going to flip it. F equals 0.45 millimeters. Okay. And then again, you have to convert that to micrometers. All right, so another question they ask you then is and you'll be doing this on the next lab, is they're gonna ask you to approximate the size. So you're gonna use this chart over and over and over again. So I have a picture here. So let's just say, for instance, this is at our 10x objective. I look on my chart, I said, oh, my field of view is 1.8 millimeters. So let's just say I counted 4 cells fit across my 1.8 diameter. In order to get the answer to that question, I'm simply going to take 1.8 and divide it by, I was doing another problem here, so I'm going to cross this out before it gets confusing. So I'm going to take my 1.8 millimeters, divide it by 4 cells, and that is going to give me 0.45 millimeters per cell, if I want to keep my units in there. Okay, so that's what you're going to do. You're going to use what you've known, and you're going to count the cells. And I was very careful on the next lab to pick slides that you could see cells and that you could kind of count them and come up with that approximate size. do be careful on your lab practical with that particular information. It will ask you to round to a specific place. So if it tells you to round to the tenths or the hundredths, make sure that you know what that means, or the nearest whole number. Okay. And then the last thing we're going to talk about is called depth of field. I was looking for a better picture, but the depth of field pictures that are online have to do with lens lengths and physics, and I was not going to get into that. So basically, we know we have our field of view. Our field of view gets smaller as magnification goes up. Okay, so we have the same thing. As you have that diameter, the amount of area you see at one point in time, there's also a depth component. So the same thing happens, okay? So you will have, and what we're saying is how much of that depth, so your slide doesn't like change in depth, it's just how much of the depth is in focus. So when you have your lower magnifications, you can see more in focus at one time than when you up your magnification. So again, the field of depth, or depth of field, gets reduced. So it's kind of like, here I have a picture. Maybe you've seen this with taking pictures. If you have, and of course many of us use our phones now, but if you had an old school camera where you had to adjust, one of the expensive ones where you focus it, you would know it depends on where the object is to what's focused. So in this case, we can see that we have this much in focus, versus here we have a lot more in focus. And it's kind of like saying, if we zoom in, we have to pick our depth we're going to focus in. You can see this when you watch movies. So when they have a widescreen lens going on, you see everything in the background, everything's nice and focused. But then they zoom in on an actor or an actress, and notice the background will get blurry. All right, so that is kind of like, depends on what's called a focal length. And that's kind of what we're talking about with our depth of field. So to demonstrate this, you would have used what we call the thread slide. Now the thread slide has 3 slides, and they're stacked on top of each other, and you can see this. All right, so right now this picture here is with the lowest, that would be with our scanning. Okay, so the question number 1 on page 19 asks, how many threads can be focused at once using the scanning objective? Now I can see all 3 of these, the red, the yellow, and the blue, are all nice and focused. Can I tell which one's on top? I think so. It looks like the blue one is on top. So if I had to put money on it, I'd say the blue one's on top and the yellow one's on the bottom. Now, when we go to the high power, that's kind of what you're seeing here. So I'm going to move my face over here. Whoop, didn't grab it. So move this over. So here's in the high power, and I wish this slide, and I just didn't see one, was a little more focused. But what you can see, or centered, is you can see that I am focused on the depth that has the yellow thread. The other threads are now no longer in focus, because then I remember our depth of field got much reduced than what we had here. So with scanning, we're able to get all 3 of those threads in focus. But since we bumped up to the high power, we're only able to get 1 at a time. So you could focus on the blue, and then the other 2 would get out of focus. You could focus on the red, and then the other 2. So you could focus only on 1 depth at a time at that high power. And I honestly cannot tell with the high power. It's very hard to tell, especially when you start focusing, which one's on top. Then it asks you which objective provides the greatest depth of field. Well, which one gives you the most amount of space at one time in focus, and that's your scanning. And then the smallest, again, would be your high power. Okay, so again, both those concepts are the same. They're inversely proportional. Magnification increases, field of view and field of depth goes down. All right, you lose that capability as you magnify. All right, so the last thing that this lab talks about is cleaning and putting away your microscope. Now, you will have noticed that there are other videos underneath this one from the lead teachers in this course, and they're going to go through all the parts, they're going to go through how to use it, and they're actually going to show you how to clean the microscope. Again, we're trying to get you prepared for future classes where you're really in the lab. So do please take time to watch these labs. All right, so that is it for lab 2. So the next thing that you need to do is you need to do the assignment on page 21 and 22. That is due Friday night, and you also need to take a picture of, I think it said the plant cell. No, I think it said the letter E for that one. So you have to take a picture, and I have the instructions in there for that 5-point. You'll see that 5-point assignment. So you also need to take a picture of what you're looking at. Just take a screenshot and then upload that as well. So again, you get 10 points for your review and then 5 points for that little home assignment that you do on your own. All right, I will talk to you later. Good luck on your assignment, and I will look forward to grading your work.